Hello, my name is Dustin Norton, and in this video we'll be explaining the breakdown of the glucose molecule to ATP. To begin, the breakdown of glucose is called glycolysis. Glycolysis involves 9 distinct reactions that convert glucose into pyruvate. The first step of glycolysis, the glucose ring, is phosphorylated. Phosphorylation is a process of adding a phosphate group to a molecule derived from ATP. As a result, at this point in glycolysis, one molecule of ATP has been consumed. The reaction occurs with the help of the enzyme hexokinase, an enzyme that catalyzes the phosphorylation of many six-membered glucose-like ring structures. The result of this phosphorylation is a molecule called glucose 6-phosphate. The next step of glycolysis includes the conversion of glucose 6-phosphate to fructose 6-phosphate. This reaction occurs with the help of the enzyme phosphoglucose isomerase. The reaction involves the rearrangement of the carbon-oxygen bond to transform the six-membered ring into a five-membered ring. In the third step of glycolysis, fructose 6-phosphate is converted to fructose 1,6-biphosphate. Similar to the reaction that occurs in step 1 of glycolysis, a second molecule of ATP provides the phosphate group that is added onto the F6P molecule. The enzyme that catalyzes this reaction is phosphofructokinase. The final step of the first stage of glycolysis utilizes the enzyme Aldolase, which catalyzes the cleavage of FBP to yield 2,3 carbon molecules. One of these molecules is called glyceraldehyde 3-phosphate, or GAP, and the other is called dihydroxyacetone phosphate. GAP is the only molecule that continues in the glycolytic pathway. As a result, all of the DHAP molecules produced are further acted on by the enzyme triphosphate isomerase, which reorganizes the DHAP into GAP so it can continue in the glycolysis. At this point in the glycolytic pathway, we have 2,3 carbon molecules, but have not yet fully converted glucose into pyruvate. In this section, we will look at the reactions that convert our 2,3 carbon molecules of glyceraldehyde 3-phosphate into pyruvate, which is the product of glycolysis. In this step, two main events take place. Glyceraldehyde 3-phosphate is oxidized by coenzyme nicotinamide adenine dinucleotide. The molecule is phosphorylated by the addition of the free phosphate group. The enzyme that catalyzes this reaction is glyceraldehyde 3-phosphate dehydrogenase. Generally, the enzyme GAPDH contains appropriate structures and holds the molecule in a conformation such that it allows the NAD molecule to pull a hydrogen off the GAP, converting the NAD to NADH. In the next step, 1,3-biphosphoglycerate is converted to 3-phosphoglycerate by the enzyme phosphoglycerate kinase. This reaction involves the loss of a phosphate group from the starting material. The phosphate is transferred to a molecule of ADP that yields our first molecule of ATP. Since we actually have two molecules of 1,3-biphosphoglycerate, we actually synthesize two molecules of ATP at this step, leaving us with a net of 0 ATP molecules up to this stage of glycolysis. The next step involves a simple rearrangement of the position of the phosphate group on a 3-phosphoglycerate molecule, making it 2-phosphoglycerate. The molecule responsible for catalyzing this reaction is called phosphoglycerate mutase. The ninth step involves the conversion of 2-phosphoglycerate to phosphoenolpyruvate, PEP. The reaction is catalyzed by the enzyme Aldolase. Aldolase works by removing a water group or dehydrating the 2-phosphoglycerate. The specificity of the enzyme pocket allows for the reaction to occur. The final step of glycolysis converts phosphoenolpyruvate into pyruvate with the help of enzyme pyruvate kinase. As the enzyme's name suggests, this reaction involves the transfer of a phosphate group. The phosphate group attached to the 2-carbon of PEP is transferred to a molecule of ADP, yielding ATP. Again, there are 2 molecules of PEP. Here we actually generate 2 ATP molecules. This process has yielded a net of 2 ATP molecules from the glycolytic pathway. After emerging from glycolysis, the 2-pyruvate are transported into the mitochondria. There, the pyruvate undergo a transaction stage before entering the actual citric acid cycle. In this phase, the pyruvate is transformed into acetyl coenzyme A, acetyl-CoA, the starting product in the citric acid cycle. The cycle begins with the reaction between acetyl-CoA and the 4-carbon oxaloacetate to form 6-carbon citric acid. Through the next steps of the cycle, 2 of the 6 carbons of the citric acid leave as carbon dioxide to ultimately yield the 4-carbon product, oxaloacetate, which is used again in the first step of the next cycle. During the 8 reactions that take place for every molecule of acetyl-CoA, the cycle produces 3 NADH and 1 FAD, FADH2, along with 1 molecule of ATP. The first reaction of the citric acid cycle is catalyzed by the enzyme citrate synthase. In this step, oxaloacetate is joined with acetyl-CoA to form citric acid. Once 2 molecules are joined, a water molecule attacks the acetyl, leading to the release of coenzyme A from the complex. The next reaction of the citric acid cycle is catalyzed by the enzyme aconitase. In the reaction, a water molecule is removed from the citric acid and then put back on in another location. The overall effect of this conversion is that the OH group is moved from the 3 to the 4 position on the molecule. The transformation yields the molecule isocitrate. 2 events occur in reaction 3 of the citric acid cycle. In the first reaction, we see our first generation of NADH from NAD. The enzyme Isocitrate Dehydrogenase catalyzes the oxidation of the OH group at the 4 position of isocitrate to yield an intermediate which has a carbon dioxide molecule removed from it to yield alpha ketoglutarate. In reaction 4 of the citric acid cycle, alpha ketoglutarate loses a carbon dioxide molecule and coenzyme A is added in its place. Decarboxylation occurs with the help of NAD, which is converted to NADH. The enzyme that catalyzes this reaction is alpha ketoglutarate dehydrogenase. The mechanism of the conversion is very similar to what occurs in the first 2 steps of pyruvate metabolism. The resulting molecule is called succinyl-CoA. Succinyl-CoA synthetase catalyzes the 5th reaction of the citric acid cycle. In the step, a molecule of guanosine triphosphate, GTP, is synthesized. GTP is a molecule that is very similar in structure and energetic properties to ATP and can be used in cells in much of the same way. GTP synthesis occurs with the addition of a free phosphate group to a GDP molecule. In this reaction, a free phosphate group first attacks the succinyl-CoA molecule, releasing the CoA. After the phosphate is attached to the molecule, it is transferred to the GDP to form GTP. The resulting product is the molecule succinate. The enzyme succinate dehydrogenase catalyzes the removal of 2 hydrogens from succinate to the 6th reaction of the citric acid cycle. In the reaction, a molecule of FAD is reduced to FADH2. The product of this reaction is fumarate. In this reaction, the enzyme fumarase catalyzes the addition of a water molecule to the fumarate in the form of an OH group to yield the molecule L-malate. The final reaction of the citric acid cycle, we regenerate oxaloacetate by oxidizing L-malate with the molecule of NAD to produce NADH. ATP synthesis is not an energetically favorable reaction. Energy is needed in order for it to occur. The energy is derived from the oxidation of NADH and FADH2 by the 4 protein complexes of the electron transport chain. The 10 NADH that enter the electron transport originate from each of the earlier processes of respiration: 2 from glycolysis, 2 from the transformation of pyruvate into acetyl-CoA, and 6 from the citric acid cycle. The 2 FADH2 originate in the citric acid cycle. The events of the electron transport chain involve NADH and FADH, which act as electron transporters as they flow through the inner membrane space. NADH is oxidized to NAD in the process. Complex 2 oxidizes FADH, garnering still more electrons for the chain. As a result of the electron transport chain, an electrochemical gradient is formed on either side of the inner mitochondrial membrane. The outside of the membrane is positive while the inside is negative. The synthesis of ATP necessitates the dissolution of the electrochemical gradient. However, since the whole process is driven by positive hydrogen ions flowing back into the matrix space from the intermembrane space, the electron transport chain maintains the electrochemical gradient by continuing to generate hydrogen ions. In total, the process started through the glycolysis of one glucose molecule yields about 32 ATP in oxidative phosphorylation. In total, oxidative phosphorylation accounts for around 90% of the body's total ATP.